Pleasant good afternoon to everyone. I want to take this opportunity to welcome our Dean, Professor Taylor, and also our Deputy Dean, Professor Felicia Stevenson, and all the persons joining us. I know Professor Roy is online and some of our presenters are online. I want to thank those who are joining us virtually and also right here. This evening is a very important evening, not just for myself, but I'm quite certain for those of you who are tuning in. It means different things for different people. For me, as a breast cancer survivor, I am here to hear what is happening with research, particularly when it comes to getting closer to a cure, and also when it comes to treatment, especially having just endured the very terrible chemotherapy treatment. So this is a very important and significant day for us at the faculty, but also in terms of looking at research. And so we have several persons who will be presenting here this afternoon. And I just want to introduce them. We have our first presenter, Mr. Stephen D'Souza, who is from the Department of Mathematics at the University of the West Indies right here. And he is currently a PhD candidate with specialization in computational nanofluid dynamics. And he will be speaking to us about magnetic cancer therapy. And it's something that I've never heard about, so I will be all ears. And then following him, we will have Dr. Regina Haraxley from the Department of the Life Sciences at the University of the West Indies, St. Augustine campus. And I'm quite honored to know that we are represented not only by our own campus here, but research is happening across all campuses. And Dr. Harrison will be presenting on precision medicine for improving breast cancer diagnostic and treatment in the Caribbean. I will be very much all ears for this one too. And just to give you a little bit of information about her, she is a Trinidadian human geneticist and lecturer in biotechnology at the University of St. Augustine at the St. Augustine campus. She leads a research group in human genomics, bioinformatics, and precision medicine, and teaches in the university's biotechnology programs. Her research focuses on developing and implementing cutting-edge technologies for studying the human genome and for discovering novel genetic contributors to various human diseases, including cancer, diabetes, and cardiovascular disease. In particular, our group is focused on applying these technologies for improving diagnostic and therapeutics in Caribbean populations for both non-communicable disease and rare disease. She received 2 bachelor's degrees from MIT in mathematics and biology, a PhD in human genetics from Yale, and postdoctoral training at Stanford in genomics and personalized medicine. Dr. Regina, we are very happy to have you, and I am looking forward to your presentation. And lastly, but of course not least, we have Professor Rupika Delgoda, and she will be presenting on tapping into nature. She is a professor of pharmacognosy, hopefully I pronounced that right. She has a DPhil at Oxford University in pharmacology and also a bachelor degree in chemistry with honors. We are very happy to have everyone on, and I'm indeed looking forward to these presentations. Without further ado, I'm going to welcome Mr. Stephen, do I call him right? D'Souza. And if you have any questions, please hold them for the end of the presentation. Please have a lot of questions also. If you want to put them in the chat, if you have to. We want to really get into the research and what is coming up from this research. So over to you.
Thank you. Good afternoon, everyone. As Doc said, I am Mr. Stephen D'Souza, a PhD candidate, and I am currently being supervised by Dr. Victor Druba and Dr. Mahesh Narayana. And I hope to do them proud today, and by extension, my department. So my talk today will be on ferrohydrodynamics, in particular applications to cancer therapy. So a bit of background information. As Doc said, we have typical treatments for cancer. One being surgery. Not sure if you can see this clearly. Is it fine? So we have surgery where we go into the tissues themselves and remove the cancerous tissue. There is chemotherapy or radiation therapy, which damages a lot of the healthy cells, it doesn't simply focus and target the cancerous cells. Although we know that it works, it has a very high mortality rating. There's also targeted therapy with radiation and so on, and drug delivery. These are some typical treatments for this cancer. So for today, I will be looking at a liver cancer because this is my current area of research. That is going to be presented later on as my final PhD seminar. So I will be looking at breast cancer, but not in this particular study. So a bit of background before I go into what exactly magnetic therapy is. So we know that the general idea for treating these cancer cells is to heat it. That's what most of the therapies are. Unfortunately, if we use radiation to heat it, what will happen is that the outer cells will be damaged first. So it will be that targeting outside and get it to the inside at a much later time. So that is a major drawback with the typical type of therapy. But as times advance, we have things called ferrofluids, which can be controlled via magnetic fields. And if we use alternating magnetic fields, what will happen is that we will cause these particles to oscillate or vibrate. And of course, relaxation time and all those things generate heat. The good thing about this type of research is that we can inject the tumor or the cancerous cells directly with this ferrofluid and use the procedure outlined before to heat. And interestingly, what will happen is that it will heat from inside outwards. So we will have a lot of damage inside of the tumor and little damage outside. That is the idea of it. Okay? So these are again typical ways we get perhaps chemotherapy via injections and so on. Some background review on what this study entailed, other studies in this area. Andros et al., I think in 2019, he studied a thermoporoelastic model for transport in tumor tissues. So the tumor was modeled as a deformable poroelastic medium, meaning that it can stretch or bend. And what they found was that the temperature, when they increased temperature within the tumor, it increased the pressure, the velocity, and the deformation within the cells. So typically in these types of research, other than Androsi, it is very limited in this type of modeling, this thermoporoelastic model. We usually assume that the cells are rigid and those that stretch. Unfortunately, that is not reality. So as a mathematician, we delve into this a bit more in detail. In my current analysis, what we are doing is looking at a cross-section of the liver tissue, where outside we have the healthy tissue cells, the omega represents the domain, and inside we have the tumor cells. Now, as you saw before, it is approximately, or usually approximately spherical. So we can use that to our advantage to get a simple domain at first. But remember that this is being modeled as a thermoelastic problem. So it will not necessarily stay as a spherical orientation. So to model this idea, we use our equations of motion, continuity equations, mass balances, and so on, to get our equations to represent what is going on. This here describes the pressure, temperature relation, as well as the deformation inside our tumor. This is our equation of mass. We have our radial deformation equation. So we have how the tissues are deforming radially, as well as axial deformation. Now, in typical cancer research, the energy equation, is not of this type. This is a new equation developed by myself and my supervisors to more accurately describe what is happening with respect to the temperature and the heating. So, and we'll come to that in a bit, but the previous models have not been necessarily very accurate when it comes to experimental data. But this equation considers a lot more things that otherwise would have been disregarded. Some important terms in that is the magnetic power dissipation, which tells you how our magnetic heating is taking place with respect to magnetic field and magnetization of the ferrofluid. We also have the relaxation time, one described by Neelian and one described by Brownian. And we have our relations to describe both here. An idea behind these, this magnetic heating is to reduce the Brownian relaxation time, but increase the Neelian, as it will cause more heating, to increase the temperature much quicker. Some thermophysical properties. For sake of time, I'm trying to stay within my 7 minutes. Thermophysical properties are given here. We have the base fluid, which is blood. We have our ferromagnetic particle, which is suspended in our base fluid to give us our ferrofluid. And we have our healthy and cancerous liver tissue. I want to bring specific attention to the heat capacities of these tissues. Notice that the healthy liver tissue has a higher heat capacity, which means that it retains more heat. So with the idea of radiation, the healthy liver tissue, since it is being heated first, will actually shield the cancerous tissue. So this is why radiation therapy takes a while to start taking effect. Also, the thermal conductivity is lower for that of the healthy tissue than that of the cancerous tissue. So again, it provides more information on how it shields the cancerous tissue from being destroyed. We also have thermophysical relations to describe our fluid. We have our density of our ferrofluid, which is a weighted average of the concentration of nanoparticles to the concentration of the fluid. Our thermal expansion is defined similarly, and the viscosity of the ferrofluid is a model taken from Sandar et al. In 2013, as it describes our ferrofluid, our blood Fe3O4 ferrofluid, much more accurately than the other models, as well as the thermal conductivity here. We will take it from that. The heat capacity, we also define it as a weighted average with the nanoparticle and the base fluid. And to solve our system, remember that we are still trying to solve this system of equations here. We have 4 equations: the continuity equation, deformation, and energy equation. So we have a system of 3 equations, very coupled, because look at this one, it has temperature, it has pressure, it has deformation. So everything is incorporated with each other. And it also goes back to what Androsi saw when he increased the temperature, he saw that pressure is increased as well. So which is what we may see from this study. So to solve our system of nonlinear differential equations, we use the finite element method with a mesh of this form. Since we assume axial symmetry, we can consider 1/2 and then get plot over 2 and get our cross section of the interested. But for sake of this talk, I will leave the regular of the finite element method out of it. Right? And we can go into a an error analysis. So to properly get our mesh, remember this is a mesh method. So we need to know what mesh will give us a low relative error when the previous mesh. So what we saw is that this mesh, 529 by 201, 529 being the number of elements, 201 being the number of time steps to a final time, gave us a relative error of less than 1%. You can ask why we go for a lower relative error. The reason being is that it takes time to run as well as computing power. So an error of less than 1% is sufficient at this point in time. If we want more accurate results or representations, we would use more advanced computing or different devices. Right? So to quantify, we must have a way, as mathematicians, we like to quantify things. We have to have a way to quantify the damage / undamaged cells. Right? So what we use is something called erroneous equation, which is used prominently in chemistry, which tells you about the fraction of damaged cells to undamaged cells. The initial concentration is given by Zeta note here, and of course this changes with space. What we expect to see is more damaged cells within the cancer tissue and less damaged cells without. So this is a paper done by Tang et al. In 2019. He also compared his results with experimental data. On the left is his results from his current study. On the right, I'm sorry, these numbers match. Yes, you can see it. Right? This is our current study here. Now, you may ask why it's here so white. It's just unfortunate that I didn't use it now. Well, so that's my error there. But we see that it is in very good relation. In fact, our results showed a 0.5% difference or relative error. The reason for this error could be because of the different models used. So since we use different models to describe the same thing, we would not get exactly the same result. But the important thing is the trend of the result. All right, the next slide was supposed to show what I got in this study, but unfortunately, it was a joke slide to bring you to my departmental seminar. The results from this study, right? So I'm teasing you. I'm just going to show you that it works, but if you wish to see further analysis, you can come to the departmental seminar about mathematics. I think it will be broadcasted. But with respect to the damage, we see here, although this is a global graph, we can extrapolate and consider what happens because this is an integral over time with temperature. So we have more damage being done here, which is the cancer area, but we see also outside the cancer area is also being damaged slightly, which is a good trade-off rather than your entire healthy tissue be damaged and the cancer tissue still being very common. Okay? So, some conclusions. I'm going to give you some conclusions on what we saw. This method of cancer treatment was very effective since we have gotten about 90% of the cancer cells dying. So with further treatments, it can be totally eradicated, or at least enough so that your immune system can start to be depressed. We have very little probability of healthy cell death, which was described by the erroneous equation. And we have a large probability of cell tissue death, which was explained previously. A very good reason for this therapy as well is that it can be controlled by external magnetic fields. So let's say that you go to the doctor and you are in the MRI machine with the curve fluids in your body. What can happen is that if it gets too hot or if it gets uncomfortable, we can take off the machine and it should reach back up to body temperature. So it can be controlled much more easily than that with the chemo, which is already in the blood. That's happening, if possible. Oh, I guess this was the joke side, right? Please come to the mathematics seminar. Scientists. So I hope to see some of you there. These are my references. Thank you for your attention and I welcome any questions at this time. Hopefully I didn't go.
Thank you so much, very interesting presentation. And today it's also breast cancer awareness. I'm aware that you said that this wasn't particularly about breast cancer, but still one of the good things that you mentioned is that the impact of it will be less, your good cells. And so I'm going to use this opportunity also to speak to the impact of chemotherapy, right? As someone who has gone through chemotherapy, my hair is coming back, but losing your hair is the least of your worries, right? It was the least of my worries. One of the impacts that it has on your body is that it brings you down to nothing, that I cannot walk from here to there without getting tired and feeling completely drained.
You know, I was just going to say, I remember that from your lecture. I attended your lecture at the overwhelming geography department. So it's about your students.
Oh, yes, so it does affect you terribly. It causes your nails to get black and dark, and your tongue it also gets dark. For some people, they lose teeth. So it's important to get your dental check. You notice I'm wearing my glasses. Now we have to wear glasses full time because without them I can hardly read. Everything is blurry. I spoke to the dark nails that over time will grow. Thank God for nail polish that we can cover that up so you can learn to live with it. And then it's a whole rebuilding process of your immune system. Because even though I fall asleep very quickly, so I don't read emails after certain hours, I'm just not up. It ages you biologically, so that even if you're in your 20s, 30s, 40s, you feel like if you're in your 60s, 70s, joint pain is another impact that I have to go. Neuropathy is another impact in terms of your feet, you say bottom feet, but I think Jamaicans say full bottom, something like that. It hurts you to even walk. So to hear about this research and to bring it back into perspective, it's important to know that we have such research taking place, because if you can get a treatment where it doesn't damage your good cell, that is the direction cancer patients would love to go. So again, thank you so much for your presentation. Continue to do good work, but if you have any question in the audience, please share, or virtually, please share your questions.
Just one quick question. What changes in your equation that it started off by saying, well, it's in a different context.
Which one?
So just any. If you were not looking at label, on breast, what would change? What would change?
All right. So let's answer that question. This is not a typical equation. It is not.
You said you have.
Right. We developed this. But what would change is not necessarily the equations, but your thermophysical properties. So as a mathematician, we generalize everything.
Okay. That's fine.
Okay.
Thank you.
No problem.
Any other questions, comments? No. You will get your official like you are here.
That's good.
Very good, very good. Very informative.
Presenter, and I want to take the opportunity right now to welcome the students and all the lecturers who will coming in. Our room is, almost out of seats, but that is good. We have our other presenter, Dr. Regine Harrison, from the Department of Life Sciences at the State University of the West Indies, St. Augustine campus. And she will be presenting on precision medicine for improving breast cancer diagnostic and treatment in the Caribbean. Let us welcome her.
Hi, everyone. Thank you for having me. I'm not, are you able to see my slides?
Not as
One
yet.
second.
Not
Sorry,
as-
1 second. Did it come up?
Yes, Doc, it's up.
Okay, great. Hi, everyone. Thank you so much for inviting me to this. And I'll start with an apology. I only learned about this like about 12 hours ago. So I'm sorry that I don't have a full-on presentation like Stefan, but I'm very happy to be here and to share a bit of the research that we've been doing involving breast cancer in my research group at St. Augustine. And I guess I'm just going to give you, Dr. Stevenson, Professor Stevenson told me 3 slides. So I'm going to give you sort of 3 windows into different approaches that we are taking to look at breast cancer diagnostics and treatments. So I'll begin by saying I'm a human geneticist, and as I'm sure you've all heard about genetic testing and the BRCA gene. And in many, developed countries, it is fairly routine now that if you have a breast cancer diagnosis, or if you have a family history of breast cancer, or if you're just a regular person who wants to know about their risk of breast cancer, you can very easily get yourself a genetic test to see if you have any known mutations that predispose you to developing a whole host of different cancers, breast cancer included. And as you know, the BRCA gene was made famous by Angelina Jolie, where she found out that she had mutations in this gene that were almost, that increased her risk for developing breast cancer, so much so that she opted for an optional double mastectomy. So the information in our genomes can tell us a lot about what our disease risk is. That is risk that you might be born with, or risk that you've inherited from someone in your family. But it can also tell us, if we look specifically at the genomes of cancer cells, it can also tell us what's driving that cancer and what's causing that cancer to continue proliferating. So there's a lot of information in the genomic and genetic data that we can get from individuals or from tumor cells in particular. So we've been focused on trying to see how we can sort of translate some of all of these advances that are now becoming so routine in the developed world into our Caribbean settings, where it is not at all routine to use genetic testing as a way of measuring breast cancer risk, or and it's also not routine to treat patient cancers based on a very precise molecular description of the patient's particular tumor. So I'm going to tell you about maybe 3 stories of different things we're doing, and I sort of crammed each one into 1 slide, but I'll talk you through it. So the first study is genetic testing in Trinidad for BRCA mutations in breast cancer patients. Now, this is not a part of our routine public health system at all. But what is routine in our public health system is that we collect preserved breast cancer biopsy tissue from patients, and that's stored for pathological purposes. So usually it's used for histology to look at the cancer under a microscope and to see what it looks like, and that determines something about how it's treated. But we can actually use that tissue to also extract DNA from those samples. And then we can test that DNA to see if it carries known BRCA mutations. And so we conducted a study in our public health system at one of our general hospitals. We took 51 samples of this of these preserved tissues. They're preserved in little blocks like what you see in the picture. And we tried to extract DNA from this. And well, we found 2 things. One is the first thing we found was that even though this can be a viable source of good quality DNA for genetic testing, we found that these particular blocks in the conditions in which they were stored, etc., were not in fact producing high quality DNA. And so it really limited the amount of genetic testing we could have done on those samples. So that's the first thing. And the second thing is, because we had such a limited amount of DNA that we were able to harness from these samples, we were only able to test for one single BRCA mutation, even though there are hundreds of known BRCA mutations. And so the quality of DNA really limited what we could have done. Now, the significance of us trying to do this initially was really just to demonstrate that we really need to improve the protocols in our public health labs for storing biopsy samples if we want to start to incorporate genetic testing as a routine pathological investigation in our samples. But more than that, we also need to test more genetic variants. We can't just test for a single BRCA mutation. And the fact that we didn't find this one mutation that we were testing for in our population does not preclude the possibility that there are other known clinically relevant mutations that are circulating in our breast cancer population, or that there are novel mutations that might be present in our Caribbean populations. Now, we tested for the most common BRCA mutation, and we didn't find it in these patients. And there have been a couple other studies as well in Trinidad, also with limited sample numbers, that tested for this BRCA mutation and didn't find it. So this brings us to, well, a hypothesis that we've had for a long time. And a second study, which we've attempted to do to kind of test this, is that we believe that in Caribbean populations, as well as other understudied populations around the world, there may be novel molecular causes of disease, such as cancer, that have not yet been discovered, because most of the studies that have been done on cancer have been done in populations from that represent a very small amount of humanity, right? Usually in developed countries. And so we then attempted to ask another question. So we know that in Trinidad, there's a small subset of patients who access private medical care, and sometimes they are able to afford to do genetic testing abroad. And if you try to do genetic testing abroad, what you would get is you would send your sample to some lab in the US or something like that, which would test for mutations that are known to be associated with breast cancer, usually in a US population. And they test a limited number of genes. Now, we looked at, we worked with a service provider in Trinidad who was facilitating this type of testing. And even though most of the patients coming into her did show a familial history of breast cancer, so you're expecting that there's something in their genes that is causing this breast cancer, only about 10% of the patients that underwent this kind of genetic testing came out with a positive test result. That is a result that showed a mutation that explained their breast cancer. And so we're asking the question, well, what about the rest of the patients who, underwent this test and did not actually find that they had a known mutation that causes breast cancer, yet they're still suffering from breast cancer. So we decided to look at some of the families that had negative test results, and we tested all of their genes, not just the few select genes that you would find in a genetic test. We looked at across their entire genome using whole exome sequencing, which is just a type of genome sequencing which sequences all of the genes in your genome, which we have about 20,000 genes. And we were looking at these genes in these families to see if we can figure out novel genetic contributors to breast cancer. And so far, we did actually find some novel potentially causative mutations in this family. And so the significance of this is that if we actually start to look, we think we might be able to discover many novel molecular causes of breast cancer that have yet to be revealed from studies in other populations. And by studying Caribbean populations and studying diverse human populations across the globe in general, we hope that we're able to sort of reveal novel biological mechanisms for disease that may not necessarily be revealed when you're only conducting studies in certain select populations. Now, this is all based sort of on the knowledge that has been sort of building up over the last 2 decades that came from the Human Genome Project. But my expertise are in human variation, human genetic variation, and we know that when you look at populations across the world, there's a huge amount of genetic variation that you're seeing across different populations. And some of that might in fact be driving genetic disease. So we think that this is the way to go in searching for that. And we have so far been successful in finding some novel things in our population. So that's about genetic testing. And then in kind of the last 2 minutes, I just want to tell you about another study that was conducted by a previous PhD student of mine, Kimberly Goodall. She's now actually a professor at University of San Francisco. So Kimberly was very interested in using clinical, demographic, and genomic data in order to be able to say something about predicting therapeutic benefits for breast cancer. So she used some data from a global database called the Cancer Genome Atlas and conducted a bunch of machine learning studies where she used many different machine learning algorithms. And she was able to actually discover a methodology using machine learning where she was able to predict with higher than 90% accuracy which patients would benefit the most from radiation therapy. And she did this using their clinical data, their demographic data, and she found that adding genomic data didn't actually make the predictions any better. So the clinical data alone and the demographic data alone were able to predict which patients were going to benefit the most from this therapy. Now, radiation therapy is grueling, and it's something that is sort of not really targeted to the patient. And so if you can, in fact, use this type of data, clinical data, which is something that we have available for all patients, right? Readily available. If you can use that to then predict the outcome of the therapy before the patient undergoes this grueling therapy, that can really help make physicians make more efficient and efficacious treatment decisions for their patients. And so we were encouraged by this result because, as you know, machine learning is making its way into medicine. And we were encouraged by this because we found that, okay, we're actually able to differentiate in our cohort which patients were going to benefit the most. So this, this was an encouraging result for us that way. So these were, and then she replicated these results in a few other different publicly available cohorts. So I just, I'll stop there, but I just wanted to let you know, these are some of the different types of investigations that we've been doing. And I know there's a lot also going on at Mona in terms of cancer research. I wanted to say thank you for organizing this forum where we could start to share a little bit of what we're doing. But I'm happy to take any questions on any of this. And if you would like to know more, all the papers are in the process of being written. I'm on sabbatical this year, so I'm hoping to get those papers finally out and published.
Thank you so much. Thank you so much, Dr. Rajini. I guess this presentation is affecting us in different ways because I want to go through genetic testing and we did not find one gene. And I remember not knowing whether to feel happy or disappointed.
Yeah.
I mean, as a breast cancer, you don't want to get the gene, but at the same time, you want to know. And I am one who still do not know. I did the genetic testing and they could not find one gene related to that. There is no breast cancer in my family. And for all cancers, there is no gene that I have in my body connected to any form of cancer. So this in itself is interesting research, and even more interesting is finding out that there are different types of breast cancer, 3 different types, and the triple negative breast cancer, which has the worst prognosis, is the one I was told that I had, and then on top of that, there's no connection. So there is one doctor who is an integrated oncologist who is considering other things outside of genes that might contribute to it. But in itself, it's good to hear that this type of research is happening, especially when you mentioned about the radiation therapy aspect of it and finding out who is more likely to respond to it. I think that is groundbreaking in and of itself, because currently they only check whether you have lymph node involved, and once you have lymph nodes involved, then you are a candidate for radiation. I did not go through radiation, but I've heard about the experience, and it's nothing nice. So this in itself is also good research and groundbreaking research, and I like the fact that it's within the context of Caribbean and all Caribbean people, because a lot of the gene testing, which is very expensive by the way, and it's not covered by insurance, I need to mention that, can cost you up to 70,000 Jamaican dollars to do it, and it's out of pocket. So that is why it's also not very popular, but it's very much necessary. So if you have any questions for Dr. Reggie, please share. I am just using the opportunity to also bring awareness while we're here. But if you do have any question, please raise your hand and let us.
One question for her, the very first slide, she spoke about the need to change public health practices for storage.
Yes, improve protocol in public health.
What kind, what kind of improvements? I'm thinking that by the time she got to the petition, the grade.
Is he asking what kind of improvements we can make?
Yes, what type?
Yes, okay. So it's been well documented that this type of biopsy tissue can be a viable source for DNA for genetic testing. It's normally stored as what we call FFPE blocks, formalin fixed paraffin embedded tissue. And as those are stored at room temperature. So you might find in the hospital that there's like a room that's just not temperature controlled, where these tissues are just stored with no special storage conditions. It has been shown that that the longer you leave the tissue stored like that, the worse the quality of the DNA that you can extract. So what I would suggest is that as these tissues are being prepared by the pathologist for their histological examination, at the same time, you can also extract either from the fresh tissue or from newly formed FFPE tissue blocks, extract the DNA right away and store the DNA, because the DNA can be stored at -20 degrees, which is a normal freezer for years and years and years. It's very stable. So improving the timeliness of DNA extraction, as well as also controlling the temperature, are two big things that can be done to ensure that these tissue blocks can actually be useful for further studies.
What do you have to do access as a researcher? Accessing for your own research? What is the process like?
To access these tissues, so we have to go through gaining ethics approval, both from UWI and then, if relevant, from the regional health authorities. And then it's actually fairly straightforward as a molecular biology technique to extract DNA from these. I had some of my undergrad and master's students help with this. And then the testing that we did is actually also fairly straightforward from a molecular biology perspective. It was PCR-based testing. And everybody has heard this term PCR after the pandemic. It's also important to know that before the pandemic, we didn't have a lot of PCR testing capability in our public health laboratories across the region. But because of the pandemic, we actually now have a lot of capability for doing DNA extraction from tissue and PCR-based testing. So this is actually something that I think, right now is something that we can actually start to implement better because we have built up that capacity over the pandemic.
So much, Dr. Rajini. It seems like we do not have any more questions. We are quite happy to have with us today, and it is excellent information. So we want to
thank you for having me.
I'm going to now ask Professor Delboda, and she will be presenting on tapping into nature. Oh, I can't wait to hear this. Yes,
Yes.
I apologize first of all for the level of preparation or the lack thereof for this presentation. I got the presentation sometime in the evening yesterday. Please arrive, speak for 7 minutes, 3 slides only. And I had multiple meetings this morning, so I just put something together and I also want to say that the experts who did the work around what I'm about to present is in the room, and that's not me. So when we have questions, I will bring that person up to answer the questions. So it's a quick summary of research that has been going on in my lab. We were the Natural Products Institute. We are currently the Guiding Center for Research in Bioscience. But the Natural Products Institute of the name suggests we were interested in nature. And so our lab was looking at natural things that have an impact on cancer cells as well as non-cancer cells. So today I just want to present a little synopsis of work that has been going on in my lab, particularly by 3 PhD students. So it's a quick summary of working. Okay. So, oops. What happened?
If you run forward, so I'm working.
Oh, okay.
Yes.
So nature has inspired man for millennia. And if you look at the type of medicines that is available now as pharmaceuticals that we go and reach to, about 32% of those have come from nature. And we know about medicines, we know that knowledge that we have gained from historical times have passed down, and scientists tap into that knowledge in order to develop drugs. If you actually look at the 140 anti-cancer agents that have been approved since 1940, over 60% can be traced to the natural product. So is this still going on? Of course. This study that was done in 2000, 20 years ago. But if you look at current data as well, nature continues to inspire us. Despite the fact that we have multiple computational abilities, the molecules that we find in nature are things that we can't quite imagine. And that's why it's so interesting for us to keep returning back to see what's inspired and what we can be more inspired by nature. So we might, there's no, yes, it's true. Hey, Christina, in glass. Cancer drugs that have come from Catarrhensis rosalis. You know this plant. You've seen this plant around campus, have you not?
Yes.
Yes, periwinkle plant. That gives very important anti-cancer drugs. It's had a song that has come from the Pacific U, a breast cancer drug that has come from that. So it's just a couple of examples of what nature has provided to us. Thank you. Okay. So can we in the Caribbean participate in this search for new cancer solutions? Well, we have 11,000 plants species, of which 72% are endemic to the region. So endemic means it's not found anywhere else. Jamaica has about a third of our myovascular plants. And then of course there's marine taxa, microbial, and natural products that are yet to be evaluated. And also we have our ethnomedical beliefs and practices. 73% of our Jamaicans rely and self-medicate on this kind of. So it is this kind of data that we look at and think there must be things in Jamaica with our ethnomedical practices that we can investigate and have them be investigated. Next slide. So Kimberly Foster, I'm taking bits and pieces from PhD thesis. Kimberly Foster, when she came and wanted to saw the fact that Jamaicans use natural products, we asked the questions, do we know whether Jamaicans use natural products for cancer treatments? We had done 3 surveys before that showed that people use it right prices for other things, but we need to know that we could identify the cancer patients. So she targeted cancer patients, and our surveys indicated that 80% of those with cancer will turn to natural products. And many times without the knowledge of our physician, and they know that because why do they do that? They believe that it works. 25% of people said that they believe that they work. And it's because it's readily available, and 40% of them said that because it's traditional. There's a very strong belief in nature. Whether they are on pharmaceuticals or not, cancer patients do turn to natural things. So it's just published on the reliance on these plant medicine by cancer patients. It's right. So then she asked the same patients, what do you use? What do you use and for what? So number 1 plant was soursop. The number 2 was guinea weed, and so on. And what did they use it for? For breast cancer, prostate cancer, and so on. And what parts did they use? Well, they used, she found that soursop used leaves and bark. So when she did the literature searches and found that there was lots of information on the leaf of soursop, but Jamaicans also used the bark. But there was no information on what was available by the bark. So she then focused on the bark, which is Jamaican soursop practices. So we took the bark and then investigated to see, well, let's see its impact on the breast and prostate cancer cells. Slide. So she prepared multiple different extracts. I think we would want to do a thorough study of those. Okay, I'm going to talk to you about 3 plants, but we are now on this one right here. Oh no, so that's what she did. No, I'm going to come back to a study quickly, but I'm starting off with Shanice's work, because of the focus on breast cancer. So we'll ask him to talk a little bit later about the things, but I'll show you what he did. But let's go to next slide. So particularly is guinea. When guinea was the second most popular, and I'm starting with this because Shanice looked at guinea for prostate cancer cells. Oh, breast cancer. Sorry. Guinea is traditionally known and has been investigated on a number of different cells, and that's literature is available. But what we didn't know was its impact on triple negative breast cancer. And so together with collaborators in the US, we looked at a number of different cell lines originating from both African American origin as well as Caucasian cell lines, triple negative breast cancer cells. And what we found was that this ingredient, dibenzyl trisulfide, this is the active ingredient that's found in henweed. So we took the extracts of henweed and looked at all of these different cells. What we found is that the DTS itself is highly potent against triple negative breast cancer. We were very excited because triple negative breast cancer is not an easy thing to treat. It's hormone resistant, and therefore coming across something that is quite potent against triple negative breast cancer was very exciting. And so that was her PhD study. I'm not going into all the details and the mechanisms that we did, but that was our guinea pig story. Next slide. Dr. Isaac Morrison looked at colon cancer. And he came to my lab with 5 particular plants that he had ideas for. He said, Prof, these are instances, this is what I'm doing. So I said, okay, look at column 5. We did his research, prepared about 60 different extracts and so on. And he found the one that was most interesting was jackfruit. So he decided to focus on jackfruit. And did a whole lot more. He had 150 different types of extracts out. And he found that the wood of the jackfruit, we couldn't unfortunately put the fruit. Technical reasons. We tried. We didn't have the electrolytes working. We just couldn't get a reliable thing that could last the entire duration. It's just necessary to prepare something like that. He started getting very good data on the lab. I'll tell you the story more because what you really find is that in the back, the wood, has this compound which is called atacapine. And atacapine is found in the fruit as well. So as he went along more and more into his study, he realized that he had a very enriched extract with high amounts of atacapine in it. And that was what he did both on the cancer cells. And we collaborated with the lab in Massachusetts to look at rats, because we don't normally do anything work by ourselves. And he found that the number of polyps that were getting formed in the colon cancer model were highly reduced, significantly reduced when he was giving the animals this atacapine rich. It's extrapolate to say that atacapine is also found in the fruit. Although we didn't get to look at the fruit itself, that this impact could be found. So it was anti-inflammatory and anti-cancer, and it was actually indicative from the enzymes that we looked at. This has cancer preventive properties. So that's Isaac's story and Chatwood's story. So we move on to the next slide, where we turn back to Kimberly. And Kimberly looked at the soursop bark, prepared extracts from the bark, and also looked at the active ingredient, which you're soon going to see, and it's called anomycin. And she looked at both the cancer cells and the normal cells. So this is the beauty of natural products in my opinion. You can see that the extract from soursop, this is 55, which means that it takes very little amount to kill the prostate cancer cells. It takes about 55 micrograms per ml to sort of have a toxic impact on the prostate cancer cell. But it requires a lot more, and she can't even quantify it properly to have an impact on the normal cells. In other words, normal cells are not impacted by the extract, but kills cancer cells. That's just what we want, right?
Yes.
of the things that we heard where you are treated, it's not selective to the cancer cells, so it will kill rapidly growing cells. That's why the hair drops off, it doesn't impact the nails, because it's killing all rapidly cells. So this was the beauty of this extract. Then she took the active ingredient, the specific active ingredient, and look at that. Really needs very little for it to begin to impact the prostate cancer cells. And again, it's much greater than that in the normal cells. Let's compare that with a known cancer drug, because cancer is a drug. So it's very small, means very potent to kill some cells very quickly. But look at that. It's the same level of the normal cells. Right? So most of the pharmaceutical drugs will impact both the cancer cells and the normal cells on an equal basis. Whereas on natural product, both the extract and the active ingredient is selective to the prostate cancer. Next slide. And I think one of the next part to be in our thesis is this. So we say, what do we do? So docetaxel is going to be given. This is the curve for its toxicity for docetaxel, which is the pharmaceutical ingredient, and it has an impact that value that you looked at. But you give the pharmaceutical along with the extract. Look what happens to the toxicity cell. It moves in the hole. In other words, the impact of docetaxel, the activity of docetaxel is 0.05 on its own, but when you give it with the extract, it becomes even more potent. So in other words, if you can give it as an adjuvant, then what it means that you don't need to give a lot of the drug. If you give the pharmaceutical drug, you have all the side effects. You can mix that with the natural product. You don't have to provide the dosage can be lowered so that the toxicity can be lowered. So as we can see, the natural products have that toxicity cells, but you can remove the toxicity of the pharmaceutical. So this is what our studies have done. Of course, these are all cellular level. It's not done on human levels or animal levels. Next slide. So, just a quick conclusion. Use your chemopreventive, that's cancer preventive and anti-cancer solutions reside in Jamaican biodiversity. And it includes terrestrial, microbial, marine. There's a lot more that we can look at. So, next slide. I thank God Lucas who have provided us funding throughout these times, and you all here. And let's keep it positive. Thank you so much. Professor
I know everyone is very excited to hear that. And amazingly, when you find out you have breast cancer, any form of cancer, you want to know what you can do. And being in the Caribbean, your mind goes natural. But I remember the first time I said to my surgeon, what is his impression of that? What he said, if you put that with your dad. Yeah. Now imagine you just scared. What your breast cancer, you hear that. It's almost, and what I found is that there are two extremes. One with nature and one with the science, and we need to see more integrative oncology happening, and we are not seeing that integration. I think it's only Dr. Williams in Bombay is the only integrated oncology. We will want to see that balance where we are integrating both the natural and also the chemotherapy and radiation and all that. But I am one who was willing to try this also. I don't know if it works. I'm just doing a trial right now. And in weed, I know I drank some cup of tea in between that, and then I learned about the turmeric and all these different things. The Taxol that I put my hair in the first 3 weeks, every bit of it. But it's good to know that we have it and that we can even, we have persons, but I think my question to you, given the fact that it is focused so much on animal study, is there an opportunity for human trials in other parts of the world? There is clinical trial with a lot of these other drugs that they are developing. And I don't know if the arts in the Caribbean, you will hear you have to travel out to be a part of those programs. Is there any opportunity in Jamaica, any part of the region, for clinical trials when it comes to using that science-based information that you are cutting at?
Yeah, I mean, I've heard of one trial that is probably ongoing, at the medical faculty on the I don't know whether it's completed yet, but we do have clinical trials unit here on campus function, which there's a really stuff to having needs that. But typically, in terms of the companies coming here asking for their products to be tested on our, but I'm aware that there was one for the industry that was happening. But I agree that it's not in the level that we want it to happen, and that's because you need ethical approval for this to happen, and often the ethics is going to look at this committee, looks at the safety profile first and foremost. And so we are basic science researchers, we do this part of the program because at least we need this information. The next level, because if everyone say, I want to test soursop team to investigate, the first thing that the committee will say is, okay, provide us with safety profile, because they can't authorize a clinical trial to take place and give people something that has not been tested for safety. That's the first thing. So there's a lot of information that we have to gather in order to do a legitimate clinical trial that is they look at what every patient is going to get and ensure that it is safe and that what the trial itself will not be toxic to the patients. And so we have to do those things in order for trials to take place.
Yes, I understand.
In agreement that ought to take this and to step into the next level.
Yeah, I understand clearly the ethics approval, and what is strange is that the clinical trials are happening informally because everybody is doing it. When we are doing it, I drink it. I don't know if it has any effect, but I have drank it and I will continue to drink it because I also love bush tea. So I grew up drinking bush tea, so it's easy for me to get into that. And so I'm very happy for this presentation in particular, and I'm quite certain all of us who are online, or you might not have, you might have family or know somebody with some form of cancer. We mentioned triple negative acid. And it's not my cancer, the cancer. It is that cancer I had, and we know out of the 3 types, it is the one with poorer prognosis because they cannot target it. They don't even know what is causing it. So to hear that it means that I'm going to drink a little more. It can observe me. I think we have a question now comment.
I do have a question. This you had mentioned something about the bark of the soursop and also the jackfruit. At what point do we know, like, is there anything been done to say what amount? How can it be used? Do we just go strip the bark off the tree and just use it?
No,
you know.
Yes,
please.
Okay.
So for the bark,
3 points here, one teaching that we noticed patients were using, so much so that when you compare which team you know,
Right.
you don't normally quantify, but
Exactly. Yes.
3 to 5 weeks and that's what the team, but for the bark is generally dry, ground up, and then they prepare like 1 cup of 2 cups.
Okay.
I just want to say,
I mean, it's usually prepared as a decoction,
Okay. Pour water. Okay.
but I mean, if you ask me, we're not saying you must go back to do that.
Yeah.
Now, studies. And then extract. So you're trying to tell us what the next step ought to be. That's what we have done to what ought to be done.
Right. Yeah.
most active, and we did it consultation wise, we saw that.
Yes, okay.
We can actually do the ground value and say that's the one that had an impact on the cancer cells. Now, that's not to say that it had an impact on
Being,
the higher concentrations and so on, but that kind of translation to take from the lab and convert it into what might happen. That's the next phase of
research, okay. Also, for the leaves, do we just pick the leaves from the tree and just put it in the pot and boil it? You know? Okay. Great. Thank you. Yes.
It's surprising, you know, because with chemotherapy, here it lists some things that must be done, but it's with chemotherapy, do you know that you have to sign something basically saying that you know that you are informed that it can kill you? Kill you, you know. Not that it can kill you, and you have to sign off on that. So it's very interesting that there are these set of steps for the natural stuff that is not even affecting your very good cells. And that is why within the world of cancer patients, I would say cancer patients and caring, there is this theory, conspiracy theory as we call it, that you know they don't want to focus on the natural because it doesn't sell, and you know people find out that they will not wait on spending the amount of money and they will lose money. Now to what extent that is true, we don't know, but within the Caribbean it's clear that we need to take charge given the fact that we have our endemic species and we have the anecdotal evidence that is saying that these things are working. And maybe if we study them a little bit more and apply them a little bit more, we will be finding cures rather than depending on drugs that even after once you have been diagnosed, it's a lifetime of watching and waiting and hoping that it does not reoccur, but it does, it can happen. So once again, thank you, Professor Delgoda. Now I am at the last part of this session where I am asked where and I'm not going to promote this opportunity to promote my book. No, we have all the science, right? And we have all the natural. And one of the things that we have not told is the spiritual. But the spiritual is very important and significant, even for those of you who are not Christians. But I can tell you there's an emotional battle that you go through when you find out that you have cancer. Very, very emotional. And for some of us, we rely strongly on God, and there are other things that we have done. Now, in my journey, right, this book is called, it's a very small book because it's deliberately small because when you're sick, you can't read nothing long. You understand? You just want something quick to motivate you and to carry on. In my journey, I started writing this book. I could not finish it, you know, because chemo was knocking me for 6 and 8 and so on. So I had to finish it at the end of the journey, which I finished chemo in March. And one of the things I tried to document in this book is some of the things that helped me mentally to deal with emotional battle of cancer. And part of it had to do not just simply with my Christianity, but also with counseling. I want to encourage people that even if you're going through something, sometimes as Black people and as Caribbean people, we don't gravitate towards counseling. We have not been grown like that. But I have a counselor that I'm still seeing, and she was very important in the journey. So I wrote this little book that I am selling at only 3,000 Jamaican dollars for anybody who wants to support the cause of breast cancer, who wants to encourage somebody, maybe you know somebody who is going through it right now. It's sad to say that I have gone through it. While I found out that I had breast cancer, my mother-in-law had found out that hers reoccurred around the same time, and she's now at stage 4, battling it very hard because it has weakened her. I had to give her imagine I have to give her my book to read during this process because there comes a time with cancer that, you know, not even science will work and you just have to rely on faith. And this book is when you have done it all. When you have done it all, you need something to build your faith. So I am here to promote it, and I thank Prof. Stevenson and Terry for allowing me this opportunity, not only to share it with us. It is good enough to be able to hear and to share You know, it's not easy to share as much as we might appear that it's easy how I love talk. It's not but even to share because you relive the experience and there's something over your head forever that you had breast cancer. It is something that is always there. We say the journey is one thing, but even after chemo, when the doctors give you up, it's like you don't want to leave, you know, because it means now you're just there waiting and watching like one morning. But at this moment, it's like you're thriving. We call it thriving after breast cancer because we have to learn to live again. Each one day at a time, and we're going to say no, and people might not like the no, it's just that we have to start protecting our health, including our mental health. So I have this little book if you need a copy. It's not signed yet, but if you want a copy, please let me know. I am at the Department of Geography and Geology. I don't even remember if I introduced myself at the beginning, and I was so busy introducing everybody else. But I'm Dr. Roseanne Smith, a lecturer in geography at the Department of Geography and Geology. So even if you do not take the book today, I am right there, right? You can inquire of me and I will get you a copy.
What's the title of the book, Doc? What is the title of the book?
Not my will, but yours with God. I don't even want to go into it. It was a very hard book to write. I never find something so hard to write it, and I love writing. But this was very difficult because it was painful to write. Very, very painful to write. And, you know, asking God, where is faith? What is faith? What does faith mean? And then coming to the reality at the very end of it all that while I want to live, and I want to be there for my 10-year-old daughter and my 5-year-old son and spend the rest of my life with my children, which brings my wonderful husband who has been a backbone and my loving children. At the end, I had no control over breast cancer. And so I reached to the conclusion, and like Jesus, that I said, God, take this cup away from me, but not my will, but yours, you know. So thank you for being here.
Is it available online, Doc?
It is, oh yes.
So what I tell you is a PR.
So while I brought in a few copies on in Jamaica, it is also available on Amazon in Kindle version for those of you who like Kindle and but for people like me who are very traditional and like to read books in my hand, it's also available in paperback. Right? So that brings me to the end of my book promotion. And now I must give people thanks. I don't know if I have any volunteer to do it. Anybody wants to volunteer? Great students. Okay. So on behalf of the Department of Physics, because they are the ones who put this together, and I really like the fact that I don't think I wasn't paying attention. Maybe I pay attention more. But I don't know if this is the first time we are having such a session or seminar during breast cancer month. But based on the information coming out from it, you see that it's a very important session for all of us. Even I want to thank Stephen. He's not here anymore. Prof was here, by the way, even though you're not seeing him. Our dean was here. I want to thank the presenters, Mr. Stephen D'Souza, Dr. Rajini Harrison, and Professor Rupika Delgoda, and our students, you know, for doing these wonderful types of research. I want to thank you for presenting it and you know, bringing awareness to the different form of cancer and letting us know what is out there and what you have been doing. I'm quite certain Grace is going to be drinking a cup of. No, and by the way, source of is also very therapeutic in terms of sleep. It's to sleep well. You know, that's what I can talk about. It does give you a good sleep like chamomile. So I'm quite certain knowing about the natural stuff, or even the gene test. If you have breast cancer in your family, I would suggest that you do the genetic testing. We are in a moment right now where you don't wait until you're 40 years old anyway. I found out my first love not at 40 years old. Right? And so I'm using this opportunity, and it does happen to male too. Breast cancer does happen to male. So I know this is something new for you. We don't want it to affect you, but I'm just bringing you into awareness. Right? But as women, one of the things I want to encourage you to do is to touch yourself when it comes to your breast and check. Right? It's for a lot of the women, I'm into breast cancer support group, and a lot of us found out not through mammogram. Some people do their yearly mammogram clear, and a few months after they feel alone, and there is breast cancer. A lot of us found alone. And I remember the first time I found alone, even after I got the ultrasound, they could not pick up that lump. And that is why I'm encouraging you to touch yourself and feel, because only you will know that if something is abnormal. I actually had to make them say, there's you can't find it. I actually had to feel it, when I found it, I said, here it is, and that is when they use the ultrasound machine and let me know that it was, it's a fibroid adenoma, it was benign. Praise God for that day. And the second time it was almost like it grew overnight. There was nothing. I went exercising, I'm there feeling pain, not like I thought it was. I exercised and I thought it was just from the exercise, but then the pain continued for days, and I touching, and there I felt a lump that turned my life upside down. And strange enough, when I did the ultrasound, the radiologist, he thought it was a fibroid adenoma. And he thought it was benign, and the results said fibroid adenoma and recommend further testing. But being the person that I was, I said, this doesn't feel, it didn't feel like it was enough, and I went to someone and said, take it out, I don't want it inside of me. And it's from the pathology of that, I found out that it was breast cancer. Early detection saves life. I would say early detection prolong lives, because with none of us know when we'll die, but at the end of the day, if you find out early, then you can get treatment. It hasn't spread to any other parts of your body, then you can get treatment. I am privileged to say that even though it was the worst type of breast cancer I found out I had, it didn't reach my lymph node, it was only within the breast area. It didn't spread to anything else, but because of safety, they know if it goes through bloodstream, you still have to do your chemotherapy, they encourage chemotherapy, but I didn't do radiation. In the last year, I've done so many scans, bone scans, CT scan, PET scans, MRI, I don't want to speak about it, and it has been a difficult journey. So my encouragement to you, even as we close this session, is not to wait on the doctors, and I don't care if you're in your 20s, because so many 20-year-olds and 20-something years old they are developing breast cancer. We don't know what is happening right now, but they are seeing it more and more to the extent they are discussing whether the mammogram should be at an earlier age. Sadly, the mammogram, because you're young and you may have dense breast, the mammogram may not pick it up, and so they have MRI. And if you're claustrophobic like me, you know that an MRI machine is not something you want to go into, but sometimes you just have a choice. So the best bet is breast cancer month. It's not just pink. We love the pink, yes, it's pretty pink, but breast cancer is a very dark journey. So my encouragement is to check yourself for yourselves, right? As much as you can. And if you haven't yet and you can take up critical illness, I encourage you also. I'm not selling insurance. I am not selling insurance, but it is expensive, right? So I'm selling my books. Because no, I can't get critical illness again, so I have to make sure that going forward, I sell something to pay for my different scans that I will have to do throughout my life. But at the same time, it's millions of dollars. The surgery alone is expensive. The chemo treatment, I have to do 16, 12 Taxol, 4 dense AC, and they call it AC red devil. And the red devil is a devil in and of itself. We try to call it different names to endure it, but I remember crying, tears, not wanting to go back. And thank God for my mother, who is still in the land of the living, who was there for me. And with her, my husband, everybody begged me, please take the other one, take it just to know they were up. And I document a lot of that in the book, but it was a tough journey and a very expensive journey. Millions of dollars type of journey. So even though I'm not selling insurance, if you can, take a critical illness. So the first time, it wasn't a lot I took, because I thought I was young and safe. So when they brought it to me, I was like, this is something, nothing, I don't have that in my family, and I'm looking at the set of disease, and I didn't think I wouldn't be standing here as a survivor of breast cancer. I didn't want any critical illness, because I'm thinking insurance and how much money they're going to get from me before I turn over and get the disease. But here I am standing today, thankful that I had some level of insurance that assisted with the expensive payments. So this is about sort of an awareness and, session, but I am urging you all to get yourself checked, check yourself, then touch them, it's yours. And also if you can, take on critical illness, and stay strong in the Lord. I mean, why not say stay strong in the Lord? Well, thanks again.
Thank you so much. And the faculty, no, thank you so much. And something the faculty is also doing is we are collecting contributions to do a contribution to the Jamaica Cancer Society. Again, doing our parts in whatever way we can to contribute to persons being treated with or prevention or management of breast cancer. Thank you for coming. Please enjoy some refreshments. And we thank those online for sticking with us. We really appreciate. Have a beautiful afternoon. You're welcome.